To understand how molecules move about our body, and especially how they move in and out of cells and across membranes, we need to understand diffusion and permeability. In this video, we will also cover how cells set up special ways to have their molecules moved according to rules of diffusion, including the molecules of the water solvent. What is diffusion? It's really a simple concept, so diffusion should not lead to confusion. Diffusion is the process by which molecules spread out across a space or a solution. Molecules are always in some random motion, usually moving about randomly with some attraction to other nearby molecules. If you had a group of 20 people standing in the middle of a football field, huddled together, and you put a blindfold on each of them and told them to start walking about in random directions, in a short amount of time, they will end up spreading out across the field. This is diffusion. If the molecules are moving randomly, the chance that they will all move in unison is very low. Diffusion is noticeable when you have a high concentration. For example, our 20 people huddled closely together. This highly concentrated pack of people, or molecules, will eventually spread out, and the resulting mixture would feature a low concentration. Remember that concentration is usually represented as an amount of solute in a certain volume of solvent. Here is another very simple example of diffusion. Placing a drop of food coloring into a glass or beaker of water. The drop itself is very highly concentrated. The concentration of the solute, the dye coloring, in that drop is very high. When this drop first hits the water, the dye molecules are still grouped together. However, due to random motion, they will slowly spread out randomly in all directions and fill the rest of the glass. We can say that the dye molecules have diffused throughout the rest of the water solution. Diffusion becomes particularly important when we talk about membranes. The difference in concentration for a particular solute between one side of a membrane and the other will determine which direction the material diffuses, and if they're moving across the membrane at all. Remember that the plasma membrane acts as a boundary between the cytoplasm and the extracellular space. And similar membranes inside of the cell can act to compartmentalize the cell further. Crossing these membranes is difficult. Molecules that move across the membranes by themselves must be soluble in water, the aqueous water environment of the extracellular space, but nonpolar enough and small enough to cross the hydrophobic part of the bilayer. Small polar or partially polar molecules can cross to a very limited extent. These membranes, though, almost completely prohibit large polar molecules and charged ions from crossing. In order for these molecules to get in or out of a cell, they must travel through special channels or gates in the membrane, which are formed by integral proteins. We will talk about two different types of movement across the membrane. The first follows the normal flow of diffusion along a concentration gradient, or, to put it another way, to diffuse from an area of high concentration to an area of low concentration. This is known either as passive transport or facilitated diffusion. other type of movement across the membrane moves molecules against a concentration gradient. This is known as active transport. Let's take these one by one. For the first example, let's look at the movement of some monosaccharides or simple sugar molecules. These molecules, depicted by green hexagons, are polar molecules, and they're too large and too polar to cross the membrane by themselves. They require a protein to form a gate or a channel for them to cross. Without this channel, the difference in concentrations between either side of the membrane will not change, since these polar monosaccharides cannot cross. But we can notice in this example that the extracellular side of the membrane has a higher concentration of the sugar compared to the cytoplasm. If we add a protein channel that is specifically shaped for a certain sugar, such as glucose, the molecules can move back and forth between the two sides of the membrane. Of course, according to what we know of diffusion, that molecules tend to move from an area of high concentration to an area of low concentration, we would expect to see the same thing occur with the sugar, and diffusion would continue until two sides have approximately equal concentration. So in this example of facilitated diffusion or passive transport, we saw a net movement of glucose into the cell. Notice here that this kind of transport falls along the natural flow for diffusion and goes from a more ordered state to a more disordered state where things are spread out randomly and evenly. This type of transport does not require an input of energy. What if the cell was consuming the glucose as it entered, or converting the glucose into something that could not flow back out? How would this affect the diffusion of glucose? Well, in this example, after the concentrations have been evened out, movement of glucose from one side of the membrane to the other still occurs, but the two sides are in a dynamic equilibrium. If we start to remove sugar from one side, we will again set up a difference in concentration, and we will again observe diffusion from one side to another. For example, if the glucose on the inside of the cell is linked together through dehydration synthesis reactions in order to form a macromolecule like starch or glycogen, then the concentration on the inside of the cell of glucose would continue to drop. Even with transport and facilitated diffusion, the concentrations will not immediately even out, because as the glucose molecules enter the cell, they would be converted to a form such as glycogen that could not be transported back out of the cell. The end result of a process like this is a flow of molecules across the membrane in one direction. This allows the cell to transport in more glucose than would otherwise be possible due to diffusion alone. Again, this is due to the ability of the cell to modify the permeability of glucose by changing it to a different form, such as glycogen. Let's look at some additional examples of facilitated diffusion or passive transport. Ions, which are atoms or molecules that carry a full positive or full negative charge, cannot pass through the nonpolar membrane. Special protein gates, known as ion channels, allow them to diffuse, again along a concentration gradient. Note that both in the previous example and in this example with ions, the solute naturally flows from high concentration to low concentration, but could only do so when the channel is open. The signal to open the channel can be based on ion concentration or even voltage, since the movement of ions and electrolytes can generate an electric charge. Sometimes these passive transporters are known as carrier proteins. They are not also all shaped like a tunnel or a channel, but sometimes even have a gate-like mechanism. In all cases, however, their overall function is the same: to facilitate the diffusion of solutes across the membrane along a concentration gradient. We can talk about different molecules and solutes like proteins, sugars, ions, and how they're transported across a membrane. Water, a small polar molecule which normally acts as the solvent for many of these molecules, also is able to diffuse across the membrane, as long as it's permeable for a particular membrane or has a transporter. This special case of diffusion, which is the diffusion of water, is known as osmosis. With solutes, it's easy to imagine how they will move. They diffuse from a high concentration to a low concentration. Water molecules behave the same way, moving from high concentration to low concentration, although here it's not as convenient to describe the concentration of water since it itself is the solvent. An easier way to remember the direction that water will diffuse is to think about the free water or the extra water in a solution or on one side of a membrane. This extra water would be the water that is not busy interacting with solutes. Therefore, on a side of a membrane with a high concentration of solutes, there will not be a lot of extra water or free water around. So this would actually represent a low concentration of free water. Water will move from an area of a high concentration of extra water to a low concentration of extra water. So we can describe this another way. We can talk about the movement of water, which is the solvent, from an area of low solute concentration to an area of high solute concentration. Remember, the area with high solute concentration will have a lot of the water busy or interacting with the solute, leaving very little free water. This can be a difficult concept to understand, but it is important to remember that osmosis is the net movement or diffusion of water across a membrane towards the side that has a higher solute concentration. In this example, there is a semi-permeable membrane in the middle of this U-shaped tube. This membrane is permeable to water, but not permeable to urea, which in this case is the solute. On one side of the U-shaped tube, the concentration of solutes is higher. Water molecules will naturally diffuse across the membrane towards the side with the higher solute concentration. So what does this mean for the cell? The membranes in the cell can be slightly permeable to water, and many cells have transporters that allow for the diffusion of water into the cell. However, since these cells contain a lot of molecules that cannot pass across the membrane, the plasma membrane for a cell is also semi-permeable, and osmosis can potentially occur. The movement of water into or out of the cell depending on which side, either extracellular or intracellular, has higher solute concentration, acts as a force or pressure on the cell. You could think of it a little bit like the pressure water places on a submarine. A cell can either be in a solution where the solute concentration on the outside of the cell is less, equal to, or more than the concentration on the inside of the cell. We have special names for these different conditions. If there are less solutes outside the cell than on the inside, the cell is in a hypotonic solution. In this case, the water will diffuse by osmosis into the cell. In the opposite scenario, a hypertonic solution, there is a higher solute concentration outside of the cell, and water will diffuse out of the cell and into the solution. Of course, if the solute concentration on the outside and the inside of the cell are balanced, there will be no net movement of water. This is known as an isotonic solution. Changes in osmotic pressure or the solute concentrations inside or outside of a cell can have drastic consequences for the life of that cell. Hypertonic solutions will cause water to leave the cell, and the cells can shrivel up and lose their shape. Perhaps even worse, cells in a hypotonic solution will fill with water and may even burst apart. Cells prefer to be in isotonic solutions, where the osmotic pressure on the cell is not a significant factor. So how do our cells keep themselves in osmotic balance with the solution? There are a number of ways that this is done. One approach is to have transporters that move water in a particular direction. Some cells even have compartments that are designed to hold excess water. This gives the cell a place to put extra water when it is placed into a hypotonic solution, and also can act as a reservoir of water when the cell encounters a hypertonic solution. Since osmosis depends on water moving towards a higher solute concentration, cells can import or export ions or other solutes to help change which side of the cell has the higher solute concentration. In our bodies, most of the cells do not have to worry too much about osmotic pressure, since our blood and interstitial fluids are normally isotonic to the rest of our cells. Of course, some cells in our body must import or export solutes in order to make sure that this fluid really is isotonic. The osmolarity of our fluids is yet another condition that our body tries to maintain at a stable homeostatic level. Another way to maintain osmotic balance is to actively pump solutes against their concentration gradient. In other words, to move molecules from a low concentration area to a high concentration area. This is known as active transport. Active transport works against a concentration gradient, and it is the opposite of normal diffusion. In order to go against the normal flow of diffusion, an input of energy is required. Usually, the chemical energy in the bonds of, let's say, a nutrient like sugar are released and directly or indirectly utilized by the cell to pump solutes against a concentration gradient. These active transport activities require a protein gate and are tightly regulated. For example, here we have a cell with a low concentration of glucose or sugar in the cytoplasm, and there is a higher concentration of glucose on the outside of the cell. This difference can be exaggerated by active transport, where chemical energy in a molecule like ATP is used to pump glucose to the outside of the cell. This can be done until there is no more sugar or no more of any other solute inside the cell. Sometimes different types of transport can be combined. In a symporter, two different molecules are moved across the membrane simultaneously. They are also moved in the same direction. In contrast, antiporters move two molecules in opposite directions. These dual transporters can combine passive transport of one molecule and use the increase in entropy from that transport to support active transport or pumping of another molecule. Here is an example of an antiporter on the left and a symporter on the right. The antiporter, in this case a sodium-potassium ion pump, uses chemical energy from ATP to move potassium ions into the cell and simultaneously move sodium ions outside of the cell. Why would the cell bother moving both ions but in opposite directions? Why not just move the sodium ions and leave potassium alone? Well, as we saw, moving sodium ions or moving solutes of any kind can potentially change the osmotic pressure experienced by the cell. By taking in potassium ions while releasing sodium ions, the cell is maintaining a similar concentration of solutes on both sides of the cell. In terms of osmosis, it doesn't matter that the actual type of solute has changed from potassium to sodium. The important part is that the relative concentration of the two combined is similar between both sides of the cell. This increased concentration of sodium on the outside of the cell, which resulted from the sodium-potassium pump, allows sodium to diffuse back into the cell through the coupled transport protein or the symporter on the right. This symporter brings sodium back into the cell along the concentration gradient and simultaneously brings glucose into the cell. This glucose can be moved against a concentration gradient because the diffusion of sodium and glucose is coupled, so as long as the concentration of both together is greater on the outside of the cell compared to the inside of the cell, then they will both diffuse in. In this way, the cell is using chemical energy from ATP indirectly to bring glucose, an important nutrient, into the cell.